hello, guys. Great to see you again. To the audience, these are a couple of guys that I started hanging out with back at COVID when it first hit, and we created a Friday night study group. And so we had a wonderful time during that whole thing talking about very, very complex biological processes and projects and whatnot. So on that note, we'll start with you, Craig. Why don't you introduce yourself and let us know, what brought you here, and then we'll jump to Leif.
Yeah, sounds good. My name is Craig Trester. I'm an applied mycologist, kind of also working in multiple fields. I got into fungi through looking at kind of the state of the world, especially with like oil spills and contamination, and thinking about like there has to be a better way. Learning that like a lot of oil remediation with the Gulf of Mexico, with Deepwater Horizon, was like spraying a petroleum product, a dispersant, which just breaks it up and is worse for the environment than petroleum. So that was kind of like an aha moment. But yeah, I've been doing a number of things with soil, mycology, microbiology, molecular biology, and it's been a very interesting trajectory where, very fitting for the soil matters. Kind of one of my focus is like how do we understand this black box beneath our feet? We understand the stars above better. And actually, Leonardo da Vinci said that. But yeah, that's how I've gotten into it. So I do a lot of consulting, teaching, research, development, and did a podcast with this guy over here as well. So we'll kind of get into more things. But yeah, we're just kind of a love of educating and sharing and finding ways with biology to really crack at a lot of these problems we have today. There's lots of solutions out there.
All right, Leif, and how about you, my friend?
Well, thanks for having me on the show, Leighton. I guess I would describe myself as a multi/interdisciplinary environmental scientist, which I guess just means I have commitment issues with what topics to actually focus on and take down in. I originally studied ecology as an undergraduate in college, kind of more community ecology, behavioral ecology, kind of like how animals organize themselves and reproduce, and a little bit into plants and how that relates to that. And then I had intermediate times of doing more of landscaping, kind of get my hands in the dirt. And then I later on got more into environmental chemistry because I started to have a fascination with the topic of bioremediation and the way that different organisms can help remove environmental pollution or regenerate ecosystems. That led me to get into environmental chemistry. I did work with wetland science because wetlands are very profound ecosystems for transforming chemicals, as well as having lots of biodiversity and applications and actually engineering applications. And then I spent time working in mycology, some for food production, some for utilizing fungi in soil restoration and other applied purposes. And now I've kind of veered a little more into anaerobic microbiology and kind of environmental microbiology in general, of another realm of organisms that can help deal with contamination problems in the environment. And and like Craig mentioned, we did a podcast together for a little while. So science communication and helping distill down these rather complex concepts into a form that people who haven't spent 10 years in various realms of the science world can hopefully understand and ultimately apply to their own practices, whatever they be.
Yeah, and we also did a couple of clubhouses years back on Future Cannabis Project.
Oh, yeah.
Where we Tom Lee in, and we really kind of did play around in that anaerobic rabbit hole, so to say.
Yeah.
Which was pretty interesting. So
similar to kind of the oh yeah, go ahead. Sorry.
No, it's all good. Very relaxed.
Yeah, no, it's similar. Well, it's so maybe kind of how we can talk about how we met, kind of like that we met initially through kind of COVID times, and set things up through like wanting to do a study group kind of with everything kind of getting shut down in detail. So kind of the running joke is like, especially in 2020, we'd have a weekly call and it would go on for like 4 hours. So kind of with the clubhouse format, getting more people in there and discuss it. And there's some recordings. And if you want to dig into some deep stuff, you can look up those cuts online. But yeah, it's something we love to share. So we'll make it a little more succinct today. So.
And I think the very first topic we did was at the study group was viruses. Are they alive or are they dead? And it might be fun to revisit that very question.
Well, I think the answer to that question is depends on how you define life, which I guess that's not that helpful, but I think it's also informative about when we look at the biological world and nature and ecosystems in general. Um, Nature doesn't necessarily lend itself to being easily defined, and so we're kind of used to hearing that these authorities, these smart people, have said this is what this means. But yeah, there's debate on terms of what life actually means. So you know viruses do a lot of the things that life do. They self-replicate, they propagate themselves. I think, from my understanding, being that I'm no expert on this topic, what makes viruses often considered not to be alive is that they have no internal metabolism. They can't like consume things and convert them into energy and then use that to sustain themselves. A virus's metabolism is more or less completely external. It's going around finding a host and using that to propagate itself. And so, I don't know, maybe that's useful for this kind of unanswerable philosophical question.
I love it. Craig, you got 2 cents on that one?
Yeah, definitely. And it's, we could dive into like the notion of what is life and kind of looking back to the history of life on this planet, going back billions of years. I think most people have heard about like the primordial soup or ooze. And a lot of the theories was that, we look at the mechanisms inside bacteria, right? We think about like the earliest organisms like a bacteria, like the ideal body form is like a pill kind of shape, and then you lead to filaments. But when you dive down into the biology internally, the molecular biology, and it's the world of polymers, of these long strands of DNA or RNA, you start kind of realizing that a lot of the functions of viruses are related to this and may have been an offshoot from a very primordial era in like the early life where it was an RNA world before a DNA world. But it's pretty amazing. So the reality is like you can look at these automatons and how they're functioning and all this amazing animations of molecular biology, but viruses may have been an offshoot of a function, maybe a mutation or an error or a parallel alternative in this RNA world that just didn't catch on quite as well, but maintained itself. And then when you look into some of the viruses that are ancient, like they're megaviruses, almost the size of the cell, and even viruses that live inside of viruses. So, maybe we'll just talk today about the bacteriophages, which are the viruses that target bacteria, which, Leif and I were talking about may have been like a really ignored part of the soil food web, because even in our bodies and across the world, they like in every surface, they play a huge role in regulating the microbial world with bacteria, which are prolific, especially with algae too. So when it comes to viruses, they're associative, but you have to think back maybe at one time, eons ago, before life was cellular, when it was more polymer, that may be an interpretation. Whereas now we have cellular life, and the polymer aspects are aspect. On our skins, we have enzymes called DNases and RNases. They're enzymes that actually break down DNA and RNA. And if you work in the lab with DNA extraction from the soil to understand microbial diversity, or RNA to understand what's being expressed in the soil, you have to protect your sample from it. You have to use molecular biology grade water. So it's interesting, right? Everything is alive and chaotic, and very much everything's an ecosystem. So that one may be a way to kind of provide some better intuition insight.
No, I love it.
It's that pure like DNA ACE, RNA ACE free water. It's like a little jug that big, and I'm making this number up, but it's very expensive. It's like hundreds of dollars for a little bottle of water that big, because they've proven that it doesn't have any of that stuff in it. But when you're talking about the early life evolution, once again, this is purely in the realm of speculation. I don't quote me on this in saying that it's actually true, but the kind of some of the hypotheses about the origins of life is that you had just RNAs that developed, and then they could replicate themselves, and then perhaps at one point some sort of protein or cellular structure encapsulated the RNA and protected it from the environment, and then it could keep going in and out. And so that's something that could have happened along the way. But let's I guess bringing it back to why this matters for soil and growing plants and things like that. This is a somewhat of an emerging field of research in terms of the ecological effect of soil viruses, because we have the soil food web kind of schematic is, you have like bacteria and fungi at the bottom, and then they're getting eaten by the soil predators, and then the plants are consuming that. But viruses are pretty abundant in the soil, and a significant percentage of bacterial cell death in the soil is caused by viruses. So with that traditional soil food web model, where it's the bacteria are there and then they're getting eaten by protozoa or nematodes and excreted, viruses attacking bacteria and then replicating inside them and bursting their cells open, this is another mechanism that leads to like nutrients leaking out of bacteria cells and becoming potentially available for plants. And like I said, this is kind of an emerging field of knowledge. A lot of like the big papers that have been written on this have only come out in the last few years. There's a research lab at University of Tennessee that is kind of one of the labs at the forefront of this. The PI's name is Mark Radesevich, I believe. But they are starting to better characterize that you have all these viruses in the soil. One interesting thing is because viruses are so much smaller than bacteria, theoretically they could be inhabiting much smaller little crevices and nooks and crannies in the three-dimensional soil architecture than bacteria can. So maybe they can be hiding out there, and then once a host organism comes by, they can pop out and start, I was about to say feeding on them, but that's the wrong term because that's not what viruses do. But ultimately, this is something that probably is really important in the nutrient cycling, and not just soil, but in all ecosystems. So it's a kind of a cool like frontier of knowledge that I had the opportunity. I was to attend a presentation online given by this researcher, and like asked, what are potential applications of this soil virus knowledge for, growing plants, improving yields and fertility? And the answer was more or less, we don't understand enough about this topic to really be able to use it for any application at this point. But we're still trying to understand it more. Maybe it will. But it makes it an exciting topic because it's something that, truly is a frontier.
Yeah, and the interesting part too is that we believe, again, theoretically, that these viruses are able to switch things on and off within the cell as well, therefore kind of pushing evolution or de-evolution, depending on which way you want to look at it. So they are a really interesting emerging understanding of the minutia of the soil food web. I mean, just the fact that, we now truly believe or understand that the bacteria can change pH in the soil. That was something that when I first started talking about that years ago, people laughed and said, that's bullshit. The biology can't shift the pH. And it's like, all right, well, what's shifting the pH in the root zone then? Why is this plant mining this material when the pH is not appropriate for that material or mineral to be released? And so maybe it goes even deeper than what we're talking about right now. And what do you guys feel about that?
Yeah, so kind of like the soil, and this is something too and this is a lot of the work that Elaine did for this advocacy, understanding the biology is critical in the soil, right? And this is coming from the understanding of like how we study soils, right? It came from geology, right? And then geological chemistry, right? If you look up an old agricultural textbook, you can grow these plants within this pH range, which means you got to add this many tons of lime, and you're basically treating it as just a growth medium, not something that's alive. So more recently, as we understood biology, it's a capital B, bio-geochemistry, right? And so, the reality is your soil is like that parent material, the sand, silt, and clay. That's the texture of your soil, right? The organic matter, right? And then also to the biology, right? So we often think about, why, like one analogy talking about how important it is to understand this connection between the biology, the organic matter, and your, the actual texture is like, why do you put lotion on your skin? Why do you like varnish a table, right? You're protecting it from oxidation, right? You're protecting that oxygen is everywhere and you can shred it. So with good soil, right, even if people will apply compost, I think one of the biggest mistakes we talked about is they'll treat it like mulch, right? They don't integrate it in, right? And what you're doing is it's not just the organic matter for the nutrient content, but it's basically a feedstock and also an inoculant with biology. And what really builds soil that holds it together are these biofilms, right? What's a biofilm? Like when you wake up in the morning and your teeth is kind of slimy, when you got kind of like, like kind of sand in your eyes or a little gummy or a little snot or like, any mucous membrane, right? But everything's an ecosystem. So one way to think of, yeah, that too. Yeah. So, and it's kind of interesting, like, and we understand these microbes live inside our gut, right? And we can think about an analogy of kind of what is a plant. It's like a gut turned inside out. Like instead of the roots on the outside, which maximize surface area that attract associated bacteria, fungi, and nematodes, protozoa, all the other organisms inside our guts, we have these microvilli. We have these intestinal linings that are filled with bacteria, filled with organisms, and then function these roles. And instead of this kind of thick, nice biofilm, when you get really good compost that smear like mayonnaise, you have mucus, right? So it's this whole interface of the organic matter, but also the state it's in, right? And then things you affect it, like with your diet or health, and much in the way you would treat it. So yeah, I think like the important thing with the soil is like this organic continuum that you maintain. And one thing we've talked about when we reduce things down, but at what point, when you walk into like an old growth forest, right, and you see how much all this biodiversity and all these different things, right, and there's lots of different ecosystems as well. They're robust and feral and kind of that way. And what you kind of ask yourself, at what point does the plant end and the soil begin? And just this continued biological matter of the plants, but even too, when you get down to the granularity of okay, it's the plant growing into the soil and all these biofilms, and there's mycorrhizal fungi that are growing around in and associated with them. Then these fungi reach out, and then those fungi even have bacteria running along them, and then those bacteria have viruses on them controlling so they don't overcome populations, and then air and water. So yeah, it's kind of like that hermetic principle, like as above is below, or in physics they call it scale invariance, right? Like a fractal, regardless of the scale, you see these similar branching patterns. But yeah, the biology is essential, and then also what the biology produces. So kind of this could be a cool thing, like where we talk about maybe like biofilms in certain ways. And I think one way we talked about it, as most people with soil, glomalin, right? This is a term that's kind of described with like glomeromycota, which are the mycorrhizal fungi. So kind of like this kind of sticky protein, iron-rich protein that will kind of bind things together. But the more research we do, we discover that it's not just the mycorrhizal fungi that do it. It's the bacteria, it's the nematodes, because everything is living together. Like the term for biofilm, or the goo that biofilm is made of, fancy word, extracellular polymeric substances, right? And so everything's exuding and exiting. And if you're familiar with soil food web, Elanium talks about as exudates, right? Things coming off it. But what are in the exudates? Sugars, proteins, amino acids, carbohydrates, the whole thing, even residues of DNA, right? Because the reality is that everything is being exuded, but in a healthy, diverse ecology, there's plenty of food for everyone. Everyone eats, right? And that's kind of what we see with succession. We disturb the land, we're resetting it. And it just wants to kind of go back in any way it can from natural disturbances, right? Yeah, that could be one way to sum it up.
No, that was awesome, because it leads right into another topic that is really cutting edge, is the understanding that horizontal gene transfer is only the tip of the iceberg. Remember the paper you boys recently about the fact that we now know for sure that the bacteria are actually sharing cytoplasm. So, I mean, that's like a whole nother level. Two different bacteria, species of bacteria come together, share cytoplasm. Now they're both unique. They're different. They're not what they were, and they're something totally new, a branch of evolution. And again, it comes back to these biofilms or relationships. Craig, what was that term? Basically, like, it was not a dimer, but when you take a core of the biofilm, there's other things that are occurring within that are outside of the cellular. Damn it! I'll have to get my head
like a strata, or like outside of the cell, like on the surface.
It was like there was like another whole realm outside of the cell, but within the biofilm.
Mm-hmm.
Well, yeah.
It's important also with biofilms to know that they have a very structural function, and so they create gradients within the biofilm. For example, there can be like a redox gradient. So like anaerobic bacteria can potentially survive in a non-anaerobic setting because they build a big biofilm where they're basically shielding themselves from oxygen that might be toxic to them. And so that's how they survive a lot of times. And with a lot of anaerobic bacteria, they'll just be floating around doing their thing until there's a stressor like oxygen, and all of a sudden, or oxygen, a lack of moisture, and now they're like, all right, now we got to like build up our bunker to protect us against that. And it's other bacteria do it too, but the biofilm has a very structural thing. It's like a way that these organisms are building a house or a fort for themselves to, survive in a more hostile environment than what would be ideal for them.
That was the term, gradient. That was exactly like the Winograd. Okay. The thicker the film is, the more different
strata.
Growths, strata are involved.
And that concept is important too with soil health. This is kind of getting a little bit of a tangent, but it's the same concept. Like soil aggregates are really important because they also create gradients. Like if you have big aggregated soil, you can have a gradient where the middle of the soil particle can have a very different redox potential, a different like electronegativity. It can have a different kind of oxygen availability than the outside of it. So if the soil is just a bunch of little broken up small little particles, it's going to be somewhat homogeneous in terms of the EH and the pH niches that exist for organisms to inhabit, or the bigger soil aggregates have that gradient as well. So in terms of all the benefits you get in the biochemistry of being able to like transfer electrons, transform nutrients, those get better and better the more that there's aggregates. And biofilms are doing a similar thing too.
Yeah. And again, people, originally we looked at biofilms as a negative. It was always antibacterial, freaking Clorox, bleach it, clean it. And in reality, we've done that to the soil. We've sprayed so much shit on it that we've broken it back into individual particulate of the sand, silt, and clay instead of having it form those aggregates, which are so important. Because again, the aggregates provide pore space for oxygen exchange as well as water, or I should say gas exchange, as well as water infiltration. And like Leif said, the middle, or even the whole thing, is in a gradient. So there's different levels all the way through into the center. So you destroy the aggregates, you reset everything back to zero. Right. And hence, guess the worst part is it collapses on itself. So now it's anaerobic.
And then even like communication and functioning, right? Signal transduction, right? We think about signals within cells or between cells, right? But we're realizing that a lot of these biofilms, especially like the different types, because we can now characterize them now, understand different composites, things. Like, for example, signaling within the cell, we're seeing similar signaling with electrical charges between biofilms or through biofilms, right? And you can kind of think about it like, think about your house, right? There's a transformer not too far away from you that is taking that high voltage line and stepping it down. And then then there's another transformer that basically goes to like your actual like electrical pole to your house, right? And then those that go into your basically your mains, and then you get your iPod, iPhone charger that's going to step it down, right? There's all that minutia as it structures, right? And so think about when you go break up the soil homogenizing, right? Imagine what life was like before there was multicellular life, right? Like we look at us, right? We have two genomes. We have our own genome, but we have also our mitochondrial genome, right? Kind of weird, right? How does that happen? It's the powerhouse of the cell. Well, like the current theory is that in the three kind of domains of life, bacteria, archaea, and eukaryota, right? Basically eukaryotes refer to like a true nucleus. The hypothesis is that there was a period of stress where these archaea, these anaerobes, would basically go and basically graze and eat other microbes, and then the mitochondria got scooped up and then incorporated. And that may have actually happened inside of a biofilm that was undisturbed, uninterrupted. There was enough stability from that environment, and there was a stressful event, competition, chemical change, right? Like the first mass extinction was the great oxygenation event, right? Where literally the sea was purple. The sea was purple because there was no oxygen, because photosynthesis hasn't evolved yet. And then when it developed, it took off and then caused this mass extinction event. So it's crazy when you think about like, how did we get to here as multicellular complex organisms, organelles and organs, all these things, from the simple aspect, the biofilm was that nice, cozy little incubator for, for geological agents that allowed that kind of to happen, right? So it is kind of, you can go pretty deep, but like the aspect too is to understand rather than to know, because you can understand a lot of intuition by understanding like kind of that kind of scale and variant in time. The more you understand something, or attempt not to know it, but to just understand it, you realize other connections, right? And our world, we live in a world of knowing, and the reason why we're having this conversation right now is because someone figured out how to engineer electrical phenomena, right? Like Faraday, he was a demonstrator of electricity in the 19th century, late 1800s, early 1900s. And he was like kind of like almost like these like big Van der Graaf things and moving things back and forth and charges. And someone asked him like, "What use is this?" And he replied, "What use is a child?" And then you had James Clerk Maxwell, who came around and wrote the Maxwell equations, wrote out equations to describe electromagnetic phenomena that allowed us to engineer and make all these devices, right? It's electrical engineering is kind of his magic. And I think we're at a similar point now with biology, with microbiology, where these microbiology phenomena that exist in the world, and we can articulate them and talk about them, but we don't have we're beginning to formalize these laws that we can observe, or not so much laws, but understandings about how things commune, right? And what's amazing is that these things have been possibly going on for as long as there's been life, like, microbial basic life. It's some of the oldest, but, it took a while to evolve an organism to have conceptualization skills to make sense of it. So the idea is like, as we push to know, I think we can come to a bigger discovery holistically by understanding as well, right? Because that's where the big advances come through when you kind of, don't say eureka. Eureka means it worked this time. Most discoveries happen when you say, that's kind of weird.
Yeah, what the fuck was that?
What was that? Let's talk about it, right? Oh, that can't happen. That's impossible, right? So that's the whole thing. Science progresses by a generation, right? So as someone who's purports to be objective in science is what we don't know, you get people that get biased by that own kind of axiom.
Yeah, it's interesting. You're talking about all that electricity stuff, and there's even bacteria out there who basically have electrical cable, like iron-reducing bacteria, which they have a metabolism where, like, oxidized iron is usually in a solid form. Think rust or orange rocks and things like that. So these bacteria actually release little cables, more or less, that then go to the solid iron and then transfer electrons to it. So they're basically shooting electricity at rocks to then change the form of the iron, and then you'd be able to use that to harvest energy. So a lot of like pretty fancy human technology is being done on a very small scale by microorganisms. And even with the biofilm communication, this is once again speculative. This is something I heard someone tell me about who's just kind of on the forefront of this research, but they're trying to identify structural proteins produced by certain bacteria in biofilms related to communication. And some of the protein structure actually looks like a barrel. It's like a tube is when they look at the shape of the protein. So it's like maybe there's a biofilm and it has a bunch of these barrel shaped proteins that are actually creating open channels that then molecules or electrons or whatever can then travel through. And another level of this kind of bacteria biofilm architecture, like they're building pretty cool stuff and they don't even need to use tools.
So they're actually building cables.
Yeah, perhaps.
To shoot information back and forth in it. I love it. I fucking love it. Yeah, and that brings up another really kind of interesting observation about back to kind of like a redox and understanding of how the sharing of subatomic particles is making things unavailable or bioavailable. This is something that we've chatted about a bunch in trying to.
I'm just gonna stop you. Not subatomic particles. It's technically. So, yeah. But so when you get subatomic, like principal quartz and things. But clarify, when a certain electrons configurations can change, right? They can make things more bioavailable. And I think one of the easiest ways people can understand that is rust, a patina, right? Iron oxide, copper oxide, right? When copper goes from orange or reddish orange to green. But yeah, it's the reality that, and these are mediated by these microbes and these enzymes. And for a long time, people thought, okay, it's one gene, one disease, one gene, one enzyme, or one thing. When in reality, it's this whole orchestration within the organism, but then with other organisms also plugged in as well. So it really is like they're the alchemists of the soil, actually.
Yeah. That's where I was trying to kind of going, was like, if you think about like a bacteria shooting a freaking lightning bolt at a chunk of iron ore to extract an electron to help it produce something else, or consume another form of that iron, and then excrete out an iron that we later used in the Iron Age. Like the whole central northern part of America is like the giant iron belt. I mean, there was so much of that material that came from, we have to believe, billions of years ago, when these organisms first started to set up shop, that we now mine and turn into steel and iron and everything else. And so, yeah, they are the alchemists of the soil. What's the matter?
Yeah, kind of on that topic, there's the big issue of acid mine drainage, which not all mine drainage is acid, but what that means is often when mining occurs in the context of you're going down into a rock formation or a mountain and basically, breaking it apart, blowing it up so you can pull out the ores and then get raw metals out of it for various, important purposes. But all those rocks are previously stable and anaerobic, and now that it's been dug up, they'll have groundwater, surface water flow through it, and then all that water and oxygen hitting those rocks allows these chemical reactions to happen, where you have things like sulfur oxidizing bacteria that are taking, a lot of the times it's like the pyrite minerals, which are like iron sulfur based minerals, but they're taking that was stably bound in the rock. Now they're oxidizing it and turning it into like a dissolved iron, which in the process produces a ton of protons or hydronium ions. So now you have this liquid coming out that's full of iron and sulfate and all these other, whatever other heavy metals or trace metals were in that material. And now they're flowing out into this water that's going to go and end up in a stream or a lake or something. And it's this global contamination issue that happens when mining occurs. It's inevitable. Figuring out how to manage that's an important kind of environmental management goal. But bring it back to the rust concept, you have this kind of reduced soluble iron that's being created by bacteria from the rock. When that water comes out of the ground, it's completely clear. It looks like normal water, even though it's loaded with iron, because the reduced iron, the iron that has more electrons on it, is soluble. So it comes out clear, but all it has to do is hit the air and it immediately starts turning bright orange. And depending on the pH, if it's a low pH, then microbial iron oxidation is really important. The pH is higher, a lot of it just happens spontaneously or abiotically, just because that reaction is energetically favored. So I know we're all into the biology here, but sometimes these profound transformations happen without it as well. So one thing about that is you can see clear water coming out of the ground, but if you get it on your pants are going to turn orange in a few minutes, because then the iron is going to oxidize and it's rust occurring. And color changes, they're a pretty profound way to see the transformations happening in the environment. So obviously there's lots of chemical reactions that don't have obvious color changes, but some of them do. And so these are kind of some of the more what would we say, gross or intuitive ways to assess what's going on with soil and rocks, is to look at the colors and see how they're changing. And so there's a lot of, I think this ties into there's a lot of tools we can have to try to figure out what's going on in a landscape that we can use without having to go take samples and send them to an analytical lab. It's not foolproof. There's multiple reasons why something might turn orange. There's multiple reasons why a soil might turn black or change colors. but we can at least use it as kind of a beginning starting point of like, aha, I see this change here. Maybe that means that this mineral has accumulated, or there's been a change in the, from being oxygenated to anaerobic. So these are, there's a lot of different things you can put in your toolkit for trying to figure out what's going on in the nature around you, and observing color changes and smells and odors. These are some of the big ones.
Yep, the nose knows. Craig, did you want to add anything to that?
Yeah, so I think one way I think a lot of people can kind of understand that, like we talked about biogeochemistry, right? Like that's principally like the fields of, chemistry, geology, and biology that kind of come together, right? And there's all those aspects. And kind of like I think one way we can see in our soils is that if you've ever kind of been somewhere in the city or development, if you've dug a foundation, you may see underneath, like the roots or the soil, this compaction layer. That usually is the case with a lot of disturbed soils. Like they're called techno soils at the technical aspect, not soils that go to a nightclub, but they technically impacted with kind of modification. But one of the things, if you go dig down there, you get down to the parent material and you'll see these thick clods, right? And these different bands of colors, right? And, even to like certain clays, which will they'll have deposits of different minerals in there. And then based upon, whether it was submerged, the microbes, other factors, you get these kind of different color iterations. But yeah, like that's one of the first ways, like understanding your soil is to approach it. Yeah, we can get at the microscope, we can get the DNA sequencer, but just going there and looking at it, understanding it, like doing a soil jar test, doing a infiltration test, a percolation test, which I'm sure Leighton has talked about. Because the reality too is you have to start somewhere on our human scale, right? Like you have to start with the geology, right? And then people look at different types of rock formations, whether you're in a sandy soil, clay, a loam, right? And there's different advantages for different types of crops. But then you have to descend down to the chemistry, which you can kind of see different factors. But the biology is something that we can really, and this is kind of a lot of the soil food concept, if you really understand and trying your best to build up the biology back through good organic matter, good processes, and doing things that are understood to be good, right? There's a lot of back and forth over anaerobes and avoiding them, but there are processes which Leighton has spoken to with, even fish manure or certain things, fermenting it and then aerobically stabilizing it and maintaining to monitor the temperature and pH, which are, factors in brewing good compost tea. We can replenish and recover it, right? I think at the end of the day is like we have to think about these things as biological interfaces. Like we I think more so like, maybe we'll think about it in the future where like it's like kind of like Neo plug into the brain thing, like that kind of thing. But in reality, like a shovel is a biological interface. Like the first stick that was used to push a furrow in a plow, a yoke that goes around an oxen to help pull that moldboard initially, right, is just much a piece of biotechnology, a biological interface as a pipette or a DNA sequencer. And then, that's the aspect. So I think to kind of people understand or getting into it is just don't be overwhelmed with how big it is. Just take it one step at a time and understand that the biology is an important component, but what you are able to interact with is using your senses, right? And the smell, the nose knows, right? If anyone's ever, left some produce out, some like brassicas soaking in water, you'll smell that hydrogen sulfide going to work. If you've left some manure sitting out or something, you smell the ammonia coming off it. And, and there's some ways where like we can allow this rotting to be good, right? Fermentation, right? What's the difference? Like, textbook like putrefaction is kind of the breakdown of proteins, but there are ways which we can steer this microbial decomposition, this good in a rotting in a good way, which can benefit us for improving the nutrition in our foods, but also making food for our plants and the soil and prepare the soil for those plants. So yeah, I think the aspect too is like to speak generally about these things. There's all this aspect, but in your journey, just kind of don't be afraid. It's one step at a time. And we have the internet, we have the Library of Alexandria in our hand, and then we have these communities. So kind of a deeper, kind of like, teleological kind of way, not getting into like the nuts and bolts, but kind of understanding like like perceive it as a trajectory where you want to learn and go to, and don't be afraid.
Yeah, and I love that you wrap the putrefaction versus fermentation, because I think that's a really important thing to understand. Both work. Like, the dairy farmers in Connecticut and up the Connecticut River Valley, they take all of their cow manure, put it in these huge lagoons that just absolutely burn your eyes when you smell them or go by them, and they apply that stuff in the spring, and it's just horrific to breathe for about a week. But it works. The corn grows and everything is fine. And then on the other side of that coin is natural farming preparations where you're actually fermenting them. They don't have that putrid smell, but they still function extremely well. And so, again, great way to put it, Craig. It's like you can't be afraid of it. I mean, yeah, you don't want to eat raw manure. The smell is there to warn your biological system, not to.
Yeah, the nose knows, right? And that's the thing about it. Like, if you've ever made pickles or a product, right? Like a good pickle, like crisp to it, right? Little bend, little crunch, a little sour. And someone figured out that if you like submerge your organic matter and you pitch a ton of salt and you create these conditions which lead to a good fermentation, right? Lactive fermentation is one example, And then if you've ever done a fermented product, oh, this kind of went off on one side, right? Oh, it's like not crunchy, it's mushy, it's kind of broken down. I didn't salt it enough, right? It's like the joke is when like, like my friend, like he's Korean, and he explained that like when his grandmother and all the other granny and his mom would get together to make kimchi, right? Like the Napa cabbage, it's like no cheating. You got to put the spice, like the spice and salt mixture between every single leaf to kind of get it thoroughly broken down. Because like these anaerobic environments, if situated in a certain way, can lead to it, right? And it's been really exciting where a lot of natural farming, a lot of Korean natural farming, a lot of Judam, really is kind of highlighted where like there's a principle. And I think even two more with Judam is really like the back like Master Cho's son. I'm not going to butcher the name. I'm just going to refer to it as that. Most people understand who I'm talking about. Had a background in like horticulture, plant science, and chemistry, and like understood that these basic principles to do it. And then even people like Nigel Palmer, I believe he was like an aerospace engineer at one point or an engineer, put together the regenerative grower guides to amendments to kind of, okay, we need to test and understand these things, right? And that, that's the thing about it. Like the same way of someone saying avoid that completely is very much the same thing as that can't happen. And, there's a reason why that happened with Elaine and like politics and different details. But if you're trying to get people on these regenerative techniques and making a lot of these inputs, farmers will just go do stuff. They'll go do stuff. They don't, even if they don't got the information right, because they don't have time to waste. So where there's a lot of nuance and a bit of a learning curve, like sometimes you want to discourage someone in a certain way, but you have to provide that reason. But it's exciting now that, especially with like, I'll just call it bio-intensive, right? Because the whole thing, KNF, natural growing, biodynamic, soul food, yada, 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 yada. They're all trying to do the same thing, right? The tribalism happens again, You just have to realize it's an entire constellation or basically galaxy of different areas basically in the same plane. So it's pretty exciting.
Leif, you want to add anything to that?
Yeah.
That was a roll.
Going back to the, when Craig was talking about the importance of just doing things and observing them, I think, just observational techniques and getting hands-on experience is extremely important. And it's something that I'll get out of touch with when I'm doing more formal science research, because I'm trying to really like understand how a system's working. I'm reading a bunch of scientific papers, analyzing data about like some process going on in the sediment. And then it gets all cerebral and conceptual. And then like some, usually I don't do it enough, but might be a few days later, weeks later, I go out to a lake or a stream, and then I'm just like digging around in the rocks and looking at things. And it's like, oh, this is where it's all happening. I can read, all the papers there are to read about like this bio-geochemical process. But if I'm never actually going and sticking my hand into the muck or walking in the muck or digging it up and seeing what it looks like, then I'll get completely out of touch with it. And it starts to become this abstract, like, what's the point type of thing. So think, no matter who you are, keeping a consistent routine of having that kind of embodied experience of not just observing, but physically interacting with the ecosystem you're working in, whether you're some, ecologist in the rainforest characterizing species, or you're a farmer running a farm interacting with your soil, or you're just, someone who has a nice hobby of composting or whatever. It's like that. Nothing can replace the hands-on primary experience of actually physically interacting with nature and, the system you're working in.
And that brings up another great point, is that, in my experience growing up as a kid, I loved playing in the swamp. But I would, step off one of these floating islands and I would sink up to my, thigh in mud and then pull my leg out and be like, whoa, holy cow, that smells like hell. Of course, I'd get home. My mother would hose me off with the hose outside, freeze my ass off. I still want that shit in the house. But that's important. That's a digestion of organic matter, and it's going to later on feed the stream or feed the, if it gets flooded, wash down and create a compost tea. So, again, all of these processes are super important. And they, as long as you understand that, I don't want to say there's nothing bad in nature, because there really isn't. It's just a process that we humans get stuck in, and we get exposed to this. It's bad. We're going to get cancer. We're going to get sick.
Yeah.
I think that's
it's kind of a thing. I think the one trend you see is that usually where there's an opportunity for an organism, it's because there's a deficiency, right? Because a lack of a community establishment, right? Like I think one paper, I think I saw in the chat, James White was mentioned, rhizophagy, but also too, the other mention was that his, some of his work, which highlighted the fact that Fusarium, Fusarium oxysporum is like a tenacious fungus, a plant pathogen, like tropical type 2 is a strain that's killing so many of the Cavendish bananas because they're all monoculture. Apparently Fusarium, when it's in the soil by itself and in the distressed soil, it'll be an opportunistic and tenacious pathogen. But it turns out some of the work was that if Pseudomonas was introduced into the soil with Fusarium, the role changes, but not changes where the fungus is now just a decomposer, but actually a mutualist, right? And we're realizing there are these bacterial fungal interactions in the soil. There's even some theories that, like we all have candida on and in our body, right? But then some people have a really gnarly candida outbreak, or, athlete's foot, like tinea pedis, what everyone has, or, a more aggressive tinea versicolor, which actually kind of blots on the skin. And so one thing we're realizing is that when you take a pretty heavy course of antibiotics, let's say you have an infection, let's say you got bit by a tick and you want to kind of knock down the aspects, you actually may be doing more harm to your internal microbial ecology, but also the microbial ecology of the fungi or other organisms in your body. And we're realizing that this dysbiosis that happens, right, this shifting towards a monoculture, have upstream effects, right? So it's funny, given the fact that we were once microbial life that then went through a symbiogenesis, right? And we crystallize our genome, right? We stabilize the number of genes we had and crystallize our metabolome, which is complexes of genes being expressed. The microbiome modulates it. It's where a lot of the general kind of theory, like the general kind of hypothesis of a lot of the microbiome theory, is not just for people, but also for microbial ecology in the soil with plants. So it's interesting that there's these up and downstream effects when you reduce to an organism by itself and view it, okay, that's a pathogen, or maybe it's just a host that is no other option, and rather than keel over and die, it's going to survive. And even too in hospitals, right? In environments where, we over sterilize, anesthetize, and it's important for an operating room and things like that. But we actually might be breeding a lot of these superbugs, because think about this. Microbes, they reproduce rapidly, right? It's the reason why Lysol says it kills 99.9% rather than 100 is because there's some genetic diversity in that population that will overcome it. And then through kind of expansion of populations from that seed, or just random DNA floating around, horizontal gene transfer, which happens all the time, bacteria that are constantly modifying themselves on every surface all the time, it's a strategy. you lead to these conditions, like for example, like Candida aureus or some really nasty antibiotic resistant bacteria. The only viable food source in an environment that is so heavily sterilized may be the macroorganism that sterilized the environment, right? So you have to understand these organisms have been on this planet for so much longer than we have. We've only been around as modern humans for like 300,000 years, and we're hubristic. We think we're hot shit. When in reality, the organisms have been on this planet for geological timeframes, right? Not ecologies, right? Geological time frame. They got a little more figured out. They're pretty robust. So it's kind of unwinding the human-centered perspective that, we are human. We can have this thought and be cognitive of it and conceptualize, but we have to be aware that, we don't exist in a vacuum. We're not islands, So it's kind of, you get a little more philosophical in that route. So,
well, it brings up a very good, interesting point, is that when you have that microbial diversity or cellular diversity, you are more suited to deal with potential external pressures in the form of these mutated cells or pathogens or whatever. And it goes back to the old understanding of, well, if you sterilize everything, the bad guys are going to get there first. So we've got to maintain that diversity and that which includes pathogens. We need those.
So this is the idea, is that you maybe want a probiotic, you want an enrichment culture, right? So this is one of the big things, I think, even with people that make cheese and dairy, right? There's a really good example. There's a really good food documentary, but it highlighted the tale that there are nuns in Connecticut, and they basically make cheese. They live a monastic life, like the convent or the monastery, right? Very simple. You make your own food, and to comply with I think the state and also FDA food production standards, they were required to use stainless steel vats for their fermentation process. And what happens is the quality of cheese changed, right? It wasn't keeping as long, and pretty much they had someone that came over from Europe and explained to them, saying, no, you need to use wood. We can't use wood. We'll get shut down. No. You want the container for your cheese to be like the inside of a cow. So wood, right? All these porous aspects. And even too, there's a lot of surface area where, if you have your favorite beer stein, right, or mug, or favorite crock, you get a biofilm on it. Those microbes move into the ceramic pores and basically establish that and protect it. It's like enrichment culture. So when you kill, right? Heck, just use a little soap. Soap's pretty great. And there's some applications like sterilizing surgical equipment. Yeah, if someone's going to be putting something in my veins or cutting me open, I kind of want it to be clean as possible. But but there are ways where we can kind of use enrichment culture to pre-inoculate, or kind of almost like a protective coating in film, which may include these biofilms that help them as well. So this is an emergent kind of property of, as we classify these different kind of roles and connections.
It's like that blue cheese, that stinky fungi.
Oh, what is it? Penicillium Roquefort?
It's something like that, yeah.
Stilton, yeah.
Leif, you want to throw something in on that one?
Yeah. Two things. First, when you're talking about if you sterilize everything, then the bad guys get there first. It also can be that you sterilize everything, you can create more bad guys, because we've talked about horizontal gene transfer a few times. And some of the types of genes that are most associated with plasmids, or kind of small little pieces of genetic information the bacteria will pass back and forth, antibiotic resistance genes are often carried on these genetic elements. So with the horizontal gene transfer, different organisms basically passing new abilities back and forth, kind of like, here's your download, here's your, new piece of software to deal with this new program. Capabilities like resisting heavy metals, resisting antibiotics, being able to break down toxins like petroleum hydrocarbons and things like that. These are all abilities that are passed between bacteria on plasmids or through other horizontal gene transfer methods. So there's actually seems to be that's the kind of thing that's favored to be able to pass along. So even organisms that may have not been antibiotic resistant, if you get into you're applying a lot of antibiotics, then ones that weren't that way, the only ones that are going to survive are going to be the ones who are antibiotic resistant. So then they're going to pick up that genetic information from others. But now, the other thing I want to say is more related to the cheese and the mug discussion. Is I was reading something about the history of mead making, which, you know, it's a very ancient alcohol brewing, fermenting practice. And, you know, back in the day, they didn't know what microbiology was, back when it was, you know, like the Vikings or the, you know, whoever, old Germanic tribes that were brewing mead. They, you know, the way it's thought is that they were like, oh, if we mix the honey and heat it up and add all this wild fruit into it, then when we stir it up, then in the middle of the night when we go to sleep, fairies will come and they'll start casting spells on this pot of liquid, and then it'll transform it into this thing that gives us all these wonderful feelings when we drink it later. And there would be, you know, these kind of family mead recipes that not only involve certain ingredients, it would involve using a specific ladle or a specific cask to make the mead. And so there's reports of, in some of these, you know, like Norse cultures, a ladle used for stirring mead would be a family heirloom that would get passed down for multiple generations. And it would be like, you have to use this ladle to stir the mead to turn it into the right type of mead. And, you know, at that time, maybe they viewed it as like, it's enchanted. It's, you know, it's been imbued with some sort of magical properties. But what probably was really going on is they're not sterilizing it. They're using the same ladle over and over. So it's building up a caked up yeast community on it. So it's like they need to use that one to mix it up because they're unbeknowingly inoculating the new batch with the right strains of yeast to ferment the right batch of mead by reusing the same thing over and over.
Love it. Fucking love it. What's that other liquid that supposedly makes you trip? It's not a Jägermeister. It's this other stuff called
absinthe. Wormwood in it.
Isn't that along the same principles, right? They're using these herbs and stuff to create an alcoholic beverage that has way more properties than just a straight whiskey or a vodka or something.
Well, yeah, that's the thing is like, you know, you have the initial fermentation process, but then you distill it down. But a lot of these complex residues, right, from plants, like if you're using whole things, for example, if you're making like a, you know, like if you're making like a wort, right, or that mash, right, you're adding in your raw ingredients, what you're pouring off, there's going to be kind of some silt. Like when you, okay, like, you know, basically you're making a whiskey by taking that initial fermented product and then distilling it again and again and again to remove the water to concentrate, right? Even to like a lot of the kind of philosophy of like the spirit, right? You know, like there's a lot of kind of goes back to alchemy and kind of like a retort or a lambic, kind of like certain kind of methods. Like you were understanding how these organic and inorganic processes were interacting, you know, through like heating and cooling, but also like these unforeseen kind of aspects, right? It's interesting because you like, you know, like when you look at like examples where like the dinoflagellates, right? These bioluminescent microbes in the Caribbean waters, right? When the Europeans came over, the Spaniards, they thought the devil was in the bay and they were shelling cannonballs. So when you splash around dinoflagellates, they grow this brilliant blue, right? You could swim through it. Even too, there's like this anecdote where a number of like during the Civil War, a number of soldiers were injured in a bog and they were crawling their way out and they're glowing because you had this bioluminescent bacteria that had kind of grown on and over them when they were in the swamp. And I'm sure the environmental quality before like heavy industry took off, like, you know, when it's still the antebellum where the South hadn't industrialized yet, I'm sure the ecology was pretty nice, right? You have a lot less things floating around and denuding. So yeah, it's kind of like these ethereal things. Yeah, we're like even too in some of Rudolf Steiner's like lectures of agriculture, and Leighton and I, you talked about this, Leif, as well. You know, if you swap out whenever he says forces, you swap out microbes. It makes a lot of sense because you're talking about this biogeochemistry. But even too, like in the lecture, his understanding of microbiology says, "Oh, I don't think they have a big role." But then he even says, like, "Oh, I may actually be wrong," because he was someone who is sitting at the foundation of these interdisciplinary fields to kind of come into as well. So yeah, it's kind of funny, like a lot of these processes which were done, and there was a narrative, a mythology, a lore that explained it. But when you track it back, it's like, oh yeah, that's like a perfect area to kind of be like, a stab, right? A stab, or even to a slant. So if anyone that's not done bacteria microbiology work, when you preserve a culture of fungus or bacteria, you'll kind of make a bit of agar in a little kind of vial or a test tube. And then, with fungi especially, you'll put in a popsicle stick, right? And the fungus doesn't really like the agar as a long-term solution. It wants to be in wood, and even longer-term culture storage for agar could be as simple as just slightly underhydrated wood shavings. So it's kind of interesting that these organisms are living in and on all these crevices, which are microscopic. I think people have an intuition with it when we talk about biochar, right? Or like pumice soil, where there's this porous microstructures which you can charge or inoculate with biology, or even soak it with different nutrients to kind of imbue or hydrate as well. So yeah, it's kind of funny, right? And you look at all the fermentation vessels, right? Wood, ceramic, right? These things are porous, right? You get into some of the people that talk about the bioceramics in Japan, right? They're porous areas that you can kind of store these materials in, and they're durable, right? They're recalcitrant. And you talk to anyone that, works in a hospital or works in microbiology research, these microbes are really hard to kill. And one of the big features is the actual structure of the material on a microscopic scale. So it's pretty amazing. This is why stainless steel is so sought after and so good, because it's a crystal, right? There are little pock marks, but there aren't these little crevices in glass. Like glass, silicon oxide. There are impurities that are unnoticeable to us, but when you get down under a macro lens or scan electron microscope, you can see that, oh wow, there's actually this. Even to, I think, one example, when I was doing mushroom cultivation, and when I was first starting, I was doing liquid culture, so doing these jars, little bioreactors of a nutrient solution, and you can use the ball jars and reuse the collars as well as the jars themselves. But the collars, sometimes they'd rust up a bit. If they rusted, I'd have to toss it, because even if I cleaned it, that rust is going to be such a higher fractal kind of surface area, which just need a little spore or a little contaminant get in there. Even if I sterilized it, they'd hang out and survive that kind of thermal shock. So yeah, you have to conceptualize, okay, when I'm looking at something, it appears to this aspect, but if I understand how it's going to look on the microbial scale, that could be a very nice little pocket to hide out in, right? If you're out in the woods, right, wilderness survival, one of the biggest things they tell you is to find an overhang of a cliff or somewhere out of the wind, right, where you can not be exposed to the elements, to keep yourself warm. Getting back to kind of the as above is below scale and variance. Imagine if you were a microbe, as if you were wandering in the woods and trying to stay warm and survive and find food. It's very similar, probably a lot more brutal on the microbial realm. Let's say that. So,
you bring up a great point about preservation. So the ancients always used clay and pottery as a way to store.
It was all they had. That was pretty much it. Yeah.
And if you think about it, in many ways, those porous surfaces on the inside was basically like a mucosal lining that was probably sealing up the ability for the oxidization coming from the outside. Because let's face it, if you put water in a clay pot, eventually that water is going to leach back out. But where you're not getting those kinds of issues in a liquid that is not perhaps as pure as water, would then actually gum up those pockets and create this protection layer from letting it escape and preserving it for thousands of years. I mean, come on, what was the thing that they found most recently? Some kind of jug of oil that they think is 5,000 years old or something. I was reading it in my feed.
Yeah, so all oils that so for example, one thing most people don't know, olive oil goes bad. So after a while, it's no longer good as a cooking oil, but it's great as a lubricant or as a lamp oil light, right? For example, olive oil goes bad because usually a lot of the organic compounds, aromatic compounds, which lead to the flavor, they go rancid, they putrefy, or maybe they'll ferment. And even too with honey, right? Understanding why honey, we can pull it out of a tomb and it's good is because it's such a super saturated solution of not only just sugar, which will basically like salt, it'll cure stuff because it'll just literally it'll strip the water out of it due to osmosis. But even too, honey is loaded with all these like proteins and different kind of plant residues and all these phytochemical residues from the process of like the bees fermenting it in their gut. And even so it's kind of crazy when you start engineering materials that may preserve things better, right? And what you lost, and that's not to say that the way we understood microbes was through pathology, through the study of disease, right? Koch kind of doing the primary doing the link between pathogen to disease through animal inoculation and histology, right? Looking at cells. Louis Pasteur with coming up with there's a certain temperature you need to heat up to resolve these different details. Like they're working within the aspect of disease, but it's been kind of an extension of like culturing them because they're pathogens and culturing them in an environment that's similar to our gut where it may be unhealthy. Versus other microbiologists at the time, like Sergei Winogradsky, he was like, okay, like this is good, but it's not an ecosystem. This is not how these microbes work all together and function, right? So when you look at the philosophies, and a lot of the amazing advancements now are using these columns to kind of simulate these environmental stresses. And I think even too, like, what's a lichen, right? There's a really good quote, I think, from Entangled Life where Merlin Sheldrake says lichens are where organisms converge into ecosystems and where ecosystems emerge into organisms or vice versa. But it's basically as a stressful event where there forces this collaboration. Like okay, like we may not always work together, but shit, like this is all we got, right? And this has happened again and again and again, right? It's like your neighbor who maybe you don't talk to too often, but if he gets broken down, he helps you out because he realizes if he gets broken down, you're helping him out, right? It's kind of we kind of, especially with COVID and kind of a lot of parasocial behavior with online and dynamics, right? Like you met people because you went outside and talked to them. You didn't have these flatland facilitating it with good intention. That's the aspect too. But yeah, it's kind of an aspect that with these biofilms and aspects, a lot of these relationships emerged by necessity. But yeah, I can, so like with lichens, think about these emergences, right? So lichen is not just a kind of frilly, brackety kind of thing, mossy looking thing on a tree, but also like we lichenized. You could even say symbiogenesis, the formation of multiple cells coming together. They were probably doing some kind of lichenization before that kind of aha moment or that event that forced that jump. So yeah, it's crazy to think about. You kind of get like very kind of philosophical or epistemological in that way, but you want to build an intuition. Intuition helps you conceptualize, right? But also too, you should think critically, but you shouldn't abandon that intuition or that under kind of deeper conceptualization for the sake of only thinking purely by logic, right?
In the case of lichen,
yeah, go ahead.
It's also it's like stress creates this relationship, but then it also leads to the emergence of kind of the superpowers in a way, because lichens are able to produce these strong organic acids that allow them to more or less eat rocks. And that's a really, really profound and important ability in an ecosystem of, soil's nice, plant's nice, it's nice to have organic matter decompose, but if there's nothing else available, you have an organism that can literally dissolve minerals out of rocks, absorb nutrients out of the air, and then grow off of rocks and air. And, that's kind of important because if you're thinking about big picture ecological succession, like how do you go from a rock to an old growth forest? It often starts with lichen because these are the organisms that can actually decompose and eat rocks. And then when they die, they've thus generated organic matter. Now, a little bit of organic matter from the lichen body can then become a substrate for early successional species, pioneer type plants to, with that little bit of organic matter, that little bit of water retention, they'll grow in the cracks of the rocks that were created by the lichen. And then they grow. And then they die and their bodies turn to organic matter. And then there's a little more soil. Now other plants that have greater nutrient requirements can grow there, and then they get bigger and they die. And then all of a sudden it builds up, and you got a whole layer of soil. And then you got plants and shrubs and trees, and eventually you have a forest or whatever the climax ecosystem is. And a lot of it derives from these symbiotic organisms that were able to survive in extremely nutrient limited, stressful environments, gaining the ability to leverage nutrients and resources out of what are otherwise very, nutrient poor diet. I'll call it that.
Hostile environments. Yeah, that's one of the reasons why I got so excited when I started working with the Protea farm. So protease are one of those that would have been right after the lichen, because it literally lives in gravel, rock, in these high UV areas.
Oh yeah.
Some of them get more moisture than others, but there's
living fossils. Living fossils, right?
Living fucking fossils. 300 million.
They figured it out. They found a good strategy, and why change, right? If it ain't broke, don't fix it. And even too with kind of ginkgo, it's kind of, you ever notice how you don't really see ginkgo trees getting eaten by insects, and they're always like pretty robust and healthy. You don't really see too much blight on a ginkgo tree, maybe aside from the aspect. There's a pretty fascinating, because they're the offshoot between where plants diverged into like, the huge variety of angiosperms and gymnosperms. For whatever reason, ginkgo were a slight offshoot. I'm paraphrasing here. There's a really excellent documentary on YouTube. It's a Japanese film about ginkgo, and the way they reproduce inside their seeds, their flowers, is akin to like how liverworts are lichens. So they make an internal little liquid sea, where literally it'll be a one gamete, and then a droplet of water would carry the gametes over. But for whatever reason, when angiosperms and gymnosperms evolved, a lot of insects chose, and a lot of the strategies that ginkgo has, they're resistant. And even too, it's used in a natural farming input as an antimicrobial or an anti-insecticide, along with like Jerusalem artichokes. So it's kind of crazy. Like when you get down to these living fossils, like look at crocodiles and alligators. They haven't changed much.
Turtles, sharks. But
turtles. Sharks are older than trees.
Right? Crazy.
What? Yeah, because then again, like life had a pretty, like, I think a pretty good, like 400 million year, or like a few 100 million year jump start in the ocean before it decided to crawl on the land.
And let's not forget the neem tree too. That's another one.
Oh yeah, it's wild. So
Cool
much
stuff.
in the same way, like these bacteria, these fungi, they're living fossils. And yeah, so that's kind of a different way to kind of look at it. It's pretty wild.
Definitely. So we've covered quite a bit of ground already. You guys, we got a little bit more time. You want to dive down a little bit more of
how much time do we have? Because I think we got some questions that some people have starred, which we
can we usually hit up questions at 1:30. So we got about 16.
Okay, cool.
To talk.
Can I say something about we mentioned glomalin a few times earlier?
Yep.
And just to get into the weeds of science here, this is something interesting I learned a few years back. Is we hear a discussion of glomalin. It's like this sticky protein-like thing that the certain mycorrhizae produce. But glomalin it's something that we call an operationally defined substance, meaning that it's not actually a real thing. It doesn't actually exist, because like you couldn't draw out like, here's the chemical structure of glomalin. It's got this many carbon rings connected. It's a substance that's defined by taking a sample, like a soil sample, and using a certain type of extraction method, where you're taking your sample and mixing it with certain chemicals with a certain pH and temperature, and then the material that comes out when you do that is glomalin. So I think this is kind of an important concept to understand, and kind of getting back into the like, nature doesn't necessarily make itself easy to define. And even though we have like terms and phrases about things, it doesn't mean you can point to like, that is that. And it's actually the same thing for humic acid, fulvic acid. Humic substances are also operationally defined. they're not real chemicals with real structures. They're things that are, you take a soil sample, and I believe it's I'll probably get this mixed up. It's either glomalin or humic acid. It's like a alkaline copper heated extraction method where it's like you're adding like a copper oxide, raising the pH to a certain level, and then heating it to a certain temperature. And whatever comes out when you do that, that's humic acid. And then there's like a follow-up size separation, and that's fulvic acid. So for a lot of the really kind of important molecules that we talk about with like building soil structure, they're purely operationally defined. And so it's interesting because I think sometimes it's easy to assume that we talk about a substance and that's like a real thing, that someone could give you a jar of like, here's a jar of pure this. And if we put it into a mass spectrometer, it'll come out and it'll have this chemical structure. But a lot of times these things are just, if we take a substance and then we add these chemicals, change the temperature and the pH, and liquid extract comes out, and that liquid extract we call this. And it kind of blew my mind when I first learned that, because I'd been using these terms for years and just assumed that they were actual, real molecules. But they're
didn't they rename it glomalin-related soil protein?
Oh,
yeah.
it's EPS.
Yeah.
Like, well, there's. So the fact is, like,
go ahead, Craig.
Yeah, that's more general. That it's more it's more a total combination. Like, yeah, I think like what could be extracted out similarly, like Sid Comlin has talked about, is like, oh, it's like an iron oxide. It's a red color. So when they were able to like, extract it, which they're doing redox chemistry on, and resolve something from it. Okay, this is like the first inkling, right? Primary research. But, as fungi have gotten more popular, and the fact like mycorrhizal fungi, soil stability and carbon sequestration, which is true, you realize, it's just this continuum of biological matter and organic matter, just like living, non-living in between and being incorporated, right? And like the turnover and uptake on the microbial realm is like pretty crazy. We think about, a lot of what we do in our body is like getting food or organic matter that we can eat ready to be digested. And even too, a lot of people that are living like very traditional lifestyles, their microbiomes help tune it in to make it digestible, right? You have, I think, some the Hadza or another kind of indigenous, a number of people in Africa that are living very close, like traditional lifestyle. They've been living for several thousand years, where they're eating these tubers from the forest, like these roots, and these things are like, it's like leather, and like if you were to eat it, you'd get like very just like you're eating like a chair. But because these people have these unique tailored microbiomes, along with cooking it, they're able to ferment and break it down more effectively. So it's like this tunability, which is really amazing. So yeah, you think about like much in the biofilm of the soil, there's the biofilm in your gut, and even too with these populations, there's, for example, one of the biggest things they talk about, what's the test, Leighton, where they measure evolution of a gas? The Braden test, the one we've talked about, because it can be problematic and if misunderstood by itself alone.
Yeah, I don't have my finger on the name, but yes, you're close. And any test.
Any test, yeah. So for example, think about this, right? And we know this in urban soils, right? I actually saw some research from some people at Urban Soils Symposium a couple years back, that urban soils, a lot of techno soils, a lot of the organic matter gets off gas. They're finding out that when it heats up, you're basically incubating the soil, and the microbes wake up and eat it up and off gas it up. But there might be some pockets due to a buildup, or even two, you need to make biofilms because it will protect the bacteriophages, which will then basically control the population, right? And what happens too is like the biofilm in the soil, these extracellular polymetric substances, all this gunk and goo, is like that incubating that protected chamber that allows them to maintain stasis or not activate too early. And we know in our gut, right? Like, why do you need fiber? Fiber is essential to feed the microbes in your gut, right? Keep like rather than just kind of to keep movements regular, they basically feed the microbes making these copulations. Turns out if you don't eat too much fiber or you're deficient in it, I think we all know like the anecdote effects of not getting enough fiber and aspect as well. But what will happen is the actual bacteria in your gut will eat away at the mucosal lining that covers your intestines. And so what prevents the bacteria from eating all the way down to the bottom, embedded in your mucosal lining, are these bacteriophages, these bacterial viruses, which will keep the population in check. The reason why the entire ocean is not covered in a green or red tide or different algae is because these viruses that kill them as quickly as they grow up. It's only in certain areas where, like in the Gulf of Mexico, or a spill where there's a sudden influx of nutrients all of a sudden, right? Like artificial aspect that they pop up, but that leads like to an anoxic dead zone because the bodies build up and tie up all the oxygen. So yeah, there's a lot of these crazy threads, right? And I think about, like I talked about earlier, like you can imagine that a gut is like a root system turned inside out or invaginated, right? Like think of all the root hairs sticking out, all the interactin points for the mycorrhizal fungi, for the decomposer fungi that's with those mycorrhizal fungi, with the rhizobium bacteria that make an infection thread and modify it, and then all the nematodes, and it's crazy to think about. There's a lot of that intuition and understanding can help kind of lead to a better granularity. But yeah, so I forgot what the original question was. I tend to do that.
Guts and gas and microbes.
Everything's an ecosystem. Everything's an ecosystem. The environment is a big gut. It's basically what a lot of
something the other day talking about gut microbes and gases, and it was like the two main types of organisms that live inside of the gut that produce gases are methanogens and the sulfate reducing bacteria. So these organisms produce the gases methane and hydrogen sulfide, and they exist in different abundances in different people's guts. Medical researchers try to tie like this bacteria is related to this disease. I wasn't getting that into it. I was more just reading, okay, so some people have more sulfide producing things in their gut. Some people have more methane producing things in their gut. Some people have more or less of both. And these are smelly gases. And maybe this was like the young, immature part of my mind started lighting up. And I was like, this is why people's farts smell different.
Yep, I'm into that. Some people.
Yeah, what is it? It's like for the gas that comes out of your stove, they have to add an odorant because the pure methane by itself doesn't smell like anything. And even too with the gasoline they put in your car, the diesel, they have to add an odorant to it. Which I kind of like the smell of. The gas smells good. I don't know. I guess when you become when you're a compost coordinator, becoming a bit of a trash sommelier, you a refined palate. Well, it's like if you ever got a truffle, right? Like an actual truffle and cut it, and you smell it. And it was funny. I was actually at this event over the weekend. It was like a kind of like a tasting menu for mushrooms, and some people that were like really into gastronomy and cooking. They made like a truffle brulee creme thing. It was great. But then we had the actual truffle, and it's like a whole truffle. Like I've only touched one like once before. They're expensive, like hundreds of dollars. Passing it around, and people are like, oh, a truffle, and they get right up in it, and they smell. They're like, oh, like it's overwhelming, right? It's kind of like that. It's I don't know, the nose knows, but there's a good funk and there's bad funk, right? It's like. I think let's like definitely pour a little out for all the homies that figured out what you can and can't eat and what you can and can't pickle, or you just not yet. We're working on it.
So you guys want to touch on phases before we get into the questions, because I'm sure a lot of the audience really doesn't get what a phase is. So maybe you can expand a little on that one.
Phases of water.
Phages.
Oh, phage. Yeah, phage. Leif, do you want to take it away? I've been running my gob a little too much. So
phages are types of viruses, right?
Yep. Phages are specifically viruses that target bacteria only, right? And and they don't affect us largely because we're multicellular. We've actually evolved mechanisms to protect us from very simple free floating RNA or DNA, which are a component largely in these viruses, right? But they target specifically from bacteria, and they control these populations. And there's a kind of cycle called the ligno, was it like a lysolytic cycle? So what happens is
Lysogenic.
that lysogenic cycle, right? So we know there are some viruses that will retroviruses that will actually incorporate their DNA into the genome of the host, right? We know this with like certain viruses like human papillomaviruses or herpes or certain retroviruses in general that you'll express it under stress, right? It's a strategy where we'll incorporate and there's many viruses that do that in general, like analogy. So for the bacteria, right? Bacteria are really kind of very simple. They have this kind of like peanut shaped, like kind of confetti little genome, this little chromatin material, and they have these like circular cassettes in the material, but it's basically a little pill, like a plastic like pill, or a pill box. And what happens is that this bacteriophage, this phage will land down. It almost looks like a moon lander. It will sit down, and it'll inject genetic information into the virus. And the phage will inject genetic information into the bacteria, and then it will hijack the natural molecular mechanisms that maintain and express genes and incorporates information. And then based upon the environment, the population density of these bacteria, right? If let's say there's a bloom, oh, this nitrogen got dumped in the water, we're going to blow up. There's a certain molecular trigger, which basically is like kind of quorum sensing, like sense who's there, that will trigger the viral element that's been incorporated into the DNA of the bacterium to then start expressing itself. And the little viruses will get made inside of the bacteria, then will burst, and they'll basically release out. And they'll either land on their viruses, maybe they'll just sit in a biofilm somewhere, but it's this natural kind of wax and wane. And you can kind of see this in similar kind of, statistical models of like populations of like rabbits over time, right? There's a certain amount of food available. And that's kind of like biostatistics, right? So statistics is like in mathematics, there are certain like just numbers, rules in the universe, just larger patterns that are there when you zoom out, right? Kind of reality, like
the lysogenic viruses, or the life cycles, in contrast to where what's called a lytic lifestyle, which is based on the word lyse. And the word lyse means to like break or rupture. So lytic, like viruses in the lytic cycle, they just infect their host and blow them up and create more viruses immediately. Where the lysogenic phase, the viruses are, like Craig said, incorporating their DNA or RNA into the host genome and just kind of staying in there for generations until it's opportune to burst them. And it's also important because we talked about horizontal gene transfer. I mentioned plasmids earlier, and then Craig also mentioned just like loose genetic material floating around the bacteria pickup. But viruses are also an important vector for horizontal gene transfer. So sometimes it's not like completely negative. It's like a virus could be transferring a beneficial trait from one bacteria to another. And they're also one of the mechanisms through which this all occurs. But with phages, they're a type of virus. I think it's important to keep in mind when we're talking about science and we get into more like complex science discussions, there's a lot of like complicated terms, and a lot of terms in science are basically equivalent to a more common word that you probably are already familiar with. And sometimes people will be like, why do they have to use so many big, confusing, complicated words? And the thing is, usually more complex terms are that way because they're more specific. And when you're getting into kind of a more nuanced discussion, these terms are useful because you can say one word instead of saying, a virus that does this and this and this. It's like we're talking about a bacteriophage, which it's funny because I've heard some like Neil deGrasse Tyson. He thinks astrophysics is the best because they use terms like giant star and blue dwarf, and he talks trash about biologists because they use complicated terms. But it's like you need to use complicated terms in biology sometimes because it reduces the number of words you can use. But I say that in the sense of like, I wouldn't be intimidated just because you hear someone using big biology words. Because if you look them up, usually it's not something that's super complicated. It's just a different language, and it's like if you're listening to someone speaking Spanish or French, what they're saying probably isn't like necessarily super sophisticated or complicated. You just don't have that vocabulary. And so looking up what the term means, it's an important step, because before I got into doing more like hands-on research in certain microbiology, I approached it from an amateur standpoint at a younger age. And I would read papers where I'm like, I know that I don't know what half of these words mean, but I'll still just read it and create a placeholder in my mind of what I think that means. And then after enough time, it starts to become more clear, like, oh, that's referring to this, even though I didn't like necessarily have the formal training at the time to understand all that. And in a way that almost improves your learning process, because if you're someone who just, goes to college and learns all the technical terminology when you're 18 and then moves on and doesn't have to go through the struggle of kind of trying to like piece it together yourself, I think there's a benefit to going through that struggle in terms of having a more like creative and innovative and, unique individualistic way of approaching the knowledge. And, while we do need in this planet, we need people who are just like the super experts who get all the formal training. We also need a lot of people who have more of the like, I came upon it in my own way and have my own kind of unique understanding, while still recognizing that, scientific authority is important. You don't want to just dismiss all the, experts and their rigid thinking just because they haven't gone through a more unique path, but all of them are important. Kind of like the importance of diversity in the soil food web and the microcosmos, and the more diversity we have in, people's capabilities and what they can think of, probably the more capable we'll be as a species of dealing with the various challenges related to preserving and creating and regenerating our environment in a way that creates a more, healthy and robust and beautiful life. So,
well put,
yeah.
Well put. And I love the creative and solution orientated approach to reading. I had a very similar experience when I first started reading white papers, and it was like after a certain time, you see that word so many times, you stop and you look it up, and then you're like, oh, now I get it. You do that repeatedly, and all of a sudden now it's crystal clear what they're trying to explain. And then what's even cooler, if you really want to go down the rabbit hole of that paper, is you look up all the sightings. So all the different sightings are all the pieces of the puzzle that led to that paper. And that too helps you really understand more about the subject, because now you understand how they got to the point where they did even discuss this topic, right? Like it's all building blocks to get there. So yeah, I try to encourage people to read white papers, but most times they'll try to read one and throw it away or give up because it is complicated.
Yeah, it's a challenge because we only have so much time in our day and so much mental capacity. But yeah, looking up a citation is important. I think it just gets into like epistemology and like, where is your knowledge coming from and how certain are you of this knowledge, which Craig kind of referenced early in our conversation of the difference between understanding and knowing, and that understanding is probably more beneficial than trying to know, because I say this from the perspective of, I've published research before that I'll get a notification that's like, your paper was cited, and I'm like, awesome, it's good for my my metrics. And then I read the paper that cited my paper, and I'm like, they shouldn't have cited my paper. What they're saying, my paper is not proving what they said there. I'm not going to complain. Give me a citation. But yeah, just because you read something in a scientific paper doesn't mean it's true or it has good evidence either. Because if you look at like, we're making this statement because we cited this paper, and then you go look up the paper it's citing, you can sometimes be like, hey, that paper didn't really say what they're suggesting it did. And it's a bit of a rabbit hole. But at the end of the day, it gives me more humility about like being careful about making definitive statements. I try to, you might have noticed me before, be like, this is speculative. Don't quote me on this. And, and actually I'll in this, with this disclaimer, don't believe what I'm saying just because I said it. If you think it's a cool idea and you want to go out and start applying it, like look it up, make sure that what I'm saying isn't bullshit. It's
agreed.
It's good
100%.
practice.
And worse
Yeah, and
yet,
and I think one big thing about is like understanding why you're doing it, right? I think one problem that I've heard a lot for some people I know that are researchers, like some students that come fresh out of undergrad, like even out of like a like even from a prestigious university, right? Um, this was even on our podcast. Damon Ty, our friend who does a lot of amazing worked for the George Genome Institute during the Human Genome Project, a lot of extracting, a lot of technical detail. It's like, oh yeah, you came from Stanford, and you don't know why we're doing this, right? Like if when you assess grades just for what's in the book, but like you don't understand like kind of the deeper conceptual process of why we're doing this thing, right? Rather than just the man.
we lost
You
you, Craig.
muted. No, he muted himself.
Oh yeah, no, but it's like yeah, like you like when you pick that up, man, I was quick on Ken. That was like, whoa.
Yeah, wasn't
But
it?
yeah, like if you're just following like a list, you're not thinking about why you're doing it, right?
Yeah,
so that's
you don't-
one big thing about it. So and that's the thing about it. If you're and then one trait I try to teach is to engage, right, and kind of tease something that's advanced on the horizon that person has to build that bridge or paddle out to get there, and kind of causes you to you have to earn it, right? You know, like, it's the whole thing about it. It's like not just like I'm going to cram for this thing and just learn it out of my head. No, like there's a process to it, and the kind of experience can be the best teacher when informed with the right theory.
So, and one other disclaimer, because I think this is important too. You'll often hear me say, this is what we know now. Because I've had so many times in my life where I've read something and it was concrete data up until that point, and then along comes a test 3 years later that proves that was bullshit, like the carbon sequestering systems. There's a number of these examples that within the last 5 years has basically racked the foundation of understanding or knowledge of what has been taught for a long period of time. It goes back to dogma and not wanting to change every textbook. And so there's a lot more to it, but just understand if you read something today, it very well may be in the near future that what that was stating was either incorrect or not complete. So again, science is a very osmotic or in motion science, for lack of better words. So we're at hour 37, one hour and 37 minutes. Ken, you want to go ahead and fire off those questions, and we'll see where it goes from there.
Sure. Dead Folk Flower has the first one here. What kind of housing should we be thinking about putting into the soil? I use pumice and biochar as an apartment for biology in my mind. Was curious if anyone has other options or insights on this.
Hell yeah. It's called compost, and in different sizes, including chips. Organic matter is a wonderful condominium. That's how I've been so successful in turning dirt into soil, was the fact that I'm taking compost, I'm washing it, breaking it apart into these small little particulate, screening and vibrating them, then putting them back in suspension in a liquid form, and then applying them. So each one of those tiny little particulates, I refer to it as chocolate water because it's so turbid you can't see through it. So that's all those little particulate that make that so you can't see through it are those condominiums or those cities. And the bigger the particulate, the more it's close to a city, where some of those super fine particulate are more like a house or a condominium. So compost and compost extracts are a wonderful vehicle or a wonderful housing technique for soil. You guys want to add anything on that?
Yeah, definitely. Like if anyone's like made compost or sifted it, your overs, right? Like the little bits of wood chips that get through in detail, take some of that and throw it in, because you're going to add like fraction of the soil. And like I mentioned earlier, like a stab or a slant, like the preferred medium for a lot of these microbes, especially fungi, is in a matrix of things, right? Especially if they're decomposers. So yeah, it's understanding that like when you dig down in the soil, it's not this perfect sifted blend that exists. There's all these different fractions, heck, of know anyone that's like had like, upstate in the Northeast, New England, like field stones, right? So many of those, a lot of the brick walls, because people were trying to clear stuff out, and you have all these rocks, but they're providing all this structure, preventing the soil from compacting, through as it went through national succession. But yeah, overs, those little bits of wood chips and stuff. You don't have to throw in the fruit stickers. Make sure you take out the twist ties or stuff like that. But yeah, like those little bits of wood chip or organic matter, they're really more your friend, because they're going to provide that diversity, because they have they're going to provide that surface area.
Leif?
Those are good answers. I don't have anything to add.
Okay, Leif, we got something going on with your microphone.
Oh, geez.
You know what? Leif, bounce and come right back. That usually fixes it.
Yeah.
Cool.
All right. Well, hopefully we answered his question.
Do you want to wait for Leif to come back for the next question?
We can start. We can start on it. Actually, I got a good answer for it, because it's actually a similar thing as well. Do you want to read it out, Ken?
Yeah, go ahead and put it up, and when here he is.
Okay.
Okay, cool.
Try your mic now.
KNF inputs. Yeah. Oh yeah.
Okay. But no, those were good answers. Those are good. I don't have anything to add.
Cool.
Can you pull that back up again, Ken? The next
Yep.
one? There we go.
I think there are some great KNF inputs, but is all that brown sugar actually natural farming?
Yeah, so that's a big kind of question in general. And that's something definitely that Jadam sought to address, because the aspect too is you have to live in an area where like sugar is produced. Sugar was like actually developed I think in Southeast Asia. That kind of grass was brought over, and yeah, it's a refining process. You need it. So ask yourself, like, how did people get sugar before there was sugar cane? Maple sugar. And even too, there's actually some history up in the Northeast about where there were people that had community guilds in making maple sugar. If you actually get your maple syrup, and you get it super hot, and you literally pour it in while it's hot, and get in like a mixer, like a bowl mixer, keep it frothing, it'll literally crystallize, right? So there's other ways to get sugar as well. But that is kind of a big critique, because you have to import it and use it, and there is some variance. There are like other ways, but there's other sugar sources. That's one example, but you need to have enough means to kind of tap trees and get enough syrup. And I don't know if anyone's ever like made sugar as well. It can kind of add it up. So beet sugar, beet sugar is made as a very industrial process. You could, and I'm even using white sugar. There's some people that have done a mix of it in general. The whole principle of it, let's understand, why are we using sugar, right? It's a curative, right? Like it's when you cure meat, when you like bacon, when you add in like the salt and sugar and brine of it, you're pulling out the water. You're basically making the microbes fight, and you're selecting for a certain range that can survive in that aspect and notion at the trade-off of presenting the organic matter, like the meat or the plant material, from kind of like totally going to mush. So there's a trade-off. So you can use different sources. But yeah, if you're in the Northeast, maple sugar, it does require a lot of fair aspects, but I feel like that's why, like a lot of this is why like Jadam, Master Cho's son, sought to kind of move past sugar, rice, because a lot of people in the world don't even access to have sugar and rice, but they got access to potatoes. There's a ton of sugar and starch in potatoes. I think even people that ain't done any kind of mushroom cultivation before and they're dirt broke, instead of going out and buying sugar, you'll just make potato dextrose. You'll just get a bunch of potatoes, chop them up, boil them, and the wort left over, the water, is loaded with sugars because it's starch sitting there. Making the sugar and crystallizing, granularizing it down, different properties, but it's understanding why you're doing it. But yeah, that is a fair question. But it's what's available in that notion, and then where it originated. Like sugar is produced in that part of the world pretty readily, and and you figure like Korea has a very similar climate to the United States, like Northeast Mid-Atlantic, but further south, like to base Southeast Asia, you got sugar, and then you go further south, the Caribbean, Cuba, Puerto Rico, sugar plantation are. So there are regional components, but it's understanding those minutia, right? That's the concept too.
Yeah,
So
you need
understanding.
You need a preservative. You need a preservative.
Yeah, yeah.
That's,
I'm not saying use salt instead.
no, because it fucks up your microbes.
Yeah, well, but even too, like fish sauce, right? Like, you know, instead of making IMO, when we talk about making fish amino acids, right? Or even too, when you look at like fermented plant juice, right? You're using the sugar, and there's it's called a chong. Like in Korea, it's literally making it's a way that they had leftover fruits. They would preserve the fruits. They would add sugar into it, and you make a syrup. And it's a fermentation kind of aspect. So it's called a chong. It's the Korean word for it. We call it fermented plant juice in English. Kind of the language barrier, you know, notion as well. But the reality is like fish, like what is it? Fish amino acids. You're adding sugar based in, but even to saltfish, right? You can render it. Even to like we're talking about fish hydrolysate or fish emulsion, right? The way people make fish sauce, and traditionally they would get a crock, they'd get some saltwater fish, and they would submerge it and keep it down. And the salt that's in the fish and the microbes in that anaerobic environment would select it. But Lord help you if that crock breaks. Holy crap. Because, you know, the way you get the oyster sauce or fish sauce is that after that process, they reduce it down. Like the way that someone makes like a super fancy, like a sauce or butter, you extract it and then boil off the water and you get a higher concentrated kind of resiny kind of goopy solution, right? Like kind of like a vinaigrette, you know, that kind of notion. So yeah, that's the short answer. Yes, sugar is a thing, but there are other ways of working with it. But also too, there are lots of different opportunities, you know, that you don't have to just use KNF alone. There's bio-intensive. It's a whole galaxy, a whole plane of all these constellations of solutions, which, you know, with the constraints to your budget or availability, you can kind of work that patchwork, right?
Yeah, I think the important thing to note is that you don't want to be using salt in these microbial preparations because you're going backwards, right? We're trying to eliminate salt in the soil solution. So again, that's why sugar was chosen, because sugar is beneficial to bacterial growth, whether it's selective or not, where salt is going to cut back your protozoa and your eukaryotes. So you don't want to go there. And Leif, did you want to add something to that as well?
Yeah, probably less practical, but taking it back to the very beginning of our conversation when you're asking about if viruses are alive, and I was like, what is life? Nature is a similar term. What is natural? What is nature? Can you define it? And it's a blurry line. So I think in this context, related to what Craig was mentioning, maybe it's like, is this truly natural versus is this an abundant resource that's available here that is inexpensive and doesn't cause damage to the environment and is sustainable and regenerative, etc. And so from like a practical perspective, maybe, you know, does it pass the purity test as being something you could just go pick out of the forest or something is not as relevant as, is it something that's cheap and abundant and not environmentally detrimental to use?
Well said.
Mm-hmm. Have you guys ever heard of people using EM, the effective microorganisms, instead of sugar? Because I've heard that that actually does work.
Yeah, so there's molasses in the EM solution. So usually when you're getting the EM by itself, like Terraganic sells it, like the regular EM, it's kind of wheat bran, molasses, and lactobacillus and yeast. And if you're getting the fancier, the Pro EM, which does, because it's a nutritive product, it's a probiotic product, they have to verify containers. That'll have the lactobacillus, the yeast, and the purple sulfur bacteria, the Rhodopseudomustris palustris. But yeah, there is some molasses in there, so there is some sugar. However, that biofilm that's made might more effectively allow to establish with minimal sugar, right? That could be another thing as well. That could allow you to use starch or the bran, right? To allow it to kind of simulate a matrix of a biofilm, which would then similarly kind of function to kind of do what that preservative way as well. Because the reality is once you get the biofilm going, they're going to be exuding other these details, right? You know, so it's a kind of thing. Even to look like a koji, right? Aspergillus oryzae, which, is used to make, soy sauce or miso. And people now are even coating meats with it, right? People are coating vegetables and fruits to kind of see how you can really pull out these flavors. And the reason why you get such a strong umami flavor from koji or soy sauce or miso is because the fungus is metabolizing the proteins to amino acids, right? The reason why, when a Parmesan cheese is little crystals, there's MSG in there. There's MSG in tomato when you reduce it. But what you're doing is it's just literally the amino acid with a sodium ion, right? It's not the ester, it's the base conjugate in that notion. But reality is you could have monosodium alanine, monosodium tryptamine, right? There's 20 proteogenic amino acids in combinations, but these are the building blocks of proteins. This is why when you taste that umami flavor, it's literally saying, hey, these are the building blocks for us. This is good. Finish it, right? It's pretty intuitive and instinctive, right? Like, why do more work? Why synthesize de novo? Right there, man. Go get them.
I want to go cook some miso soup for dinner now.
Right?
Ooh, nice.
Well, we'll jump back into the questions here. Let's see. Where did the shrub seeds come from? Now, this was a conversation in the chat where they were trying to figure out where, you've got.
I think their thing was, well, what came first, the plant or the seed?
Well, yeah, they got it in the chicken and the egg, and
that's where we ended up with a mid-succession shrub, but it would go all the way back to the lichen. So the lichen had to start to allow for plants or soil building, so plants could evolve into the soil, connect with more microbes that were evolving in the water, coming ashore, and creating these successionary plant systems. So
I'm not holding the whole evolutionary timeline of all the domains of life in my mind. I should study more so I can. But if you're talking about like a rock face, they could be coming from bird poop. Is one place. The animals, wind, there's different vectors to get them around.
But when it's back in the lichen, and that's all there was before there was birds, etc. That's where they're coming. Like, where the hell did this come from? But then you look at quorum sensing and creating biology that doesn't exist in the soil system by the biology getting together and figuring it out and creating it.
Well, it goes back to what's first, chicken or the egg. Well, it was obviously the chicken was a mutated version of the parent bird because the bird ate something funny or fucked a different bird or whatever.
Over millions of generations. That's you just like, because I just think back to there's who was that one, it was like you've seen the crocodile, right? It was like very on the nose, hand wavy, like, if evolution's real, where's the duck crocodile hybrid? But it's not. You need to stretch the time frame out. But if you're viewing Earth is only 5,000 years old,
where it is. It's called the platypus.
Well, yeah. Well, it's just why? Just because that worked. Okay, cool, neat. It's
But I'm putting my money on the egg coming first because there had to have been some bird that was very similar to a chicken, but not technically a chicken. But then it laid an egg that had whatever genetic recombination that whatever hatched out of that was a chicken. So I think I don't think the other way works. I think it has to be that the egg came first.
Well, of course, for that species. But when you go back all the way, it's like, all right, well, what was it that caused the ability for that first bird to lay the first egg? Well, that goes, wow, down the hole you go. So lichen. It all comes from lichen.
Birds came from the
dinosaurs. You have to have an environment, that is advantageous, right? Like the amniotic, right? Think about the biofilm. You have to have all those nutrients where it's warm and incubated, right? And that aspect, even too with seeds, right? When seeds land in the ground, right? It turns out that drying seeds may not be the best. That it might be best to store seeds in the soil where they fall. and that right temperature and humidity in general, because there's even microbes that transfer, vertically inside the seed that are in the parent and get incorporated into the actual germ, and these are plant growth promoters, things like that. But yeah, I think the idea to wind it back, it's like, sex, right? Sex was useful because you can make more novelty, right? Because previously it was about just, asexually reproducing, right? There's animals like trypanosomes. They're crazy. They're kind of like a weird, they're like a weird protozoa nematode thing, and they can reproduce sexually and asexually. And the way they do it, they divide like the top. They're like a Y thing, but then when they have sex, like the female or equivalent trypanosome makes a cleft, and the male slides into it, and it's like kind of gross. But yeah, but the thing is, in an environment, the more genetically this is a very clinical explanation, my good sir. Is just like I can make anything unsexy if I wanted to. Like I can expound on it, just make it clinical in that aspect. No, but the reality is like why? Like so, okay, what necessity was there for the egg, right? If you could just make a copy of yourself, right? Oh, well, maybe more diversity to fight against viruses, right? More chance to make these new features, right? So it's that whole aspect. Well, think about this, like what was before birds? Well, the hypothesis that the birds evolved from dinosaurs or small reptiles that survived the extinction event, right? And the theory is that we came from mammals that could hide and survive the extinction event, right? What was before that?
More important, platypus, mammal that lays an egg.
Yeah. Like, well, there you go. That's the thing. There was a time where it's like, oh man, crap. Okay, so I can't, if I have a litter of pups, I can't swim away as fast from predators, but I can lay them. You go back, right? That's the whole thing. It's the pressures of the environment do that, but that's the best. So what was before the dinosaurs, right? Like what crawled out of the sea, like a fish thing, right?
Yeah.
And then
what started things in the sea? Maybe it was the genetic code that started in the water that landed on our planet and created the beginning of life. Totally.
Well, yeah. That's the thing about it too. Is not even that. Pan polymeria, that polymers, the primary details. It might have been that Earth was the best condition that these organic compounds floating around asteroids hit. The whole primordial ooze, the experiment where they conduct and shock it, and you get in those aspects. It's chaos. That's the thing about it. We like to think that things are very linear and clean cut. No, it's just chaos.
Sure.
And was the genetic code for that chicken actually in the water already when it landed here? So it was just waiting for the right components and the right circumstances to come together to create the first chicken.
Well, to get there. Well, here's a crazy thing. So in chickens, we can turn on genes that will turn their beak into teeth. Yeah,
so,
because we see similarities in reptiles.
If they start breathing fire, we're really in trouble. Okay, Leighton was trying to get something out. Leighton, what did you have to say?
Thankfully, there's not enough oxygen in the atmosphere for that. If there was enough oxygen in the atmosphere to breathe fire like that, we'd have millipedes that are 9 or 12 feet long. There'd be other problems to worry about, and also just lighting a cigarette might be a bit more of an affair. So that's that also too. Especially with all this organic matter floating around.
All I was trying to say is, an egg is just biofilm dried out.
Yeah.
An evolutionary process. They laid blobs. That didn't work, so they let the blobs dry in the sun, and that created the shell that prevented the other pests from getting to their
duration, right?
Yep.
So,
and that's just So it's. I think we have one last one. It's directed for me. I like vermiculite. What are Craig's thoughts on it? So vermiculite, I think, it's useful, but I think a lot of people are not the biggest fan of it because it's mined from a certain source and it's finite. But it's useful, right? If you have it in your shed or you go to a garden center and it's on sale, like, sure, but also do there's other options as well. Like, I think some people in the kind of like regenerative or organic community, because it is it's like azomite, right? Azomite's a mineral source mined from one area where it's composite, but if you apply azomite continuously, you can get that nutrient lock in some ways. Even too, I think with biochar, you can over add biochar, right? Don't be a moron. Yeah, don't add it on. So, but yeah, like, it's not verboten, like, oh, thou shalt not use vermiculite. If you got it and it's around and it's sitting around, it's like, I bought a bag of gypsum for like a 40-pound bag of gypsum for like adding in a little gypsum into my cultures for mushroom cultivation or my agar. I've had that same 40-pound bag of gypsum for the past like 8 years because I only use a little bit at a time, right? But it depends, right? But I think if you're growing and using a lot of it, there are other alternatives to it. I know some people use like biochar or other kind of ways to bring it in, larger particles, charcoal. But, it's about being aware what you got, right? Just, nothing should be like we have the internet. You have the Library of Alexandria in your pocket. Pretty wild. It's like you need Google, you can search. And then heck now, like you even have AI, which you can help you search and resolve things, right? It's not the end of the world. We're pretty creative. We got thumbs and social skills. That's why we're here today.
Thumbs up. Yeah. Leif,
thumbs up, man.
You haven't said anything much, so.
Oh, yeah. It's a lot of conversation here. Just to go on that same concept, plastic is really useful sometimes. I don't try to use it if I don't need it, but like sometimes it's like, I want a plastic bag for this function. And if I try to use something else, it's not going to work. Or doing lab work, I know people who are like, oh, I hate wasting all these pipette tips. But I'm like, if I reuse this pipette tip and contaminate a culture and then have to restart a whole experiment, that little bit of plastic was probably worth it to use there. And so it's cost benefit analyses and risk assessments and things like that are how we should think about this. And, I think being a purist of like, we're going to move past using any of this ever can be a little bit, small-minded and, not
pedantic
picture.
Yeah. Well, it's like, plastic, great for holding nuclear isotopes, storing samples, maybe not the best thing to put your water in for drinking or like your sandwich that you're going to eat and like be done with. Yeah. Been using natural materials for a while, so.
Cool.
Well, guys, we're actually at the 2 hours.
Oh, cool. Groovy.
Yeah, I was going to say thank you guys so much for coming on today and audience and people that listen to this in the future. That's why these guys are my study group. We can cover some crazy fucking distances between topics and subjects and really enjoy the conversation. So I really appreciate you guys giving me 2 hours today. Leif, I know you got a shit ton of work coming up in the future. So go get them, Tiger, and appreciate that you give us this time today. And Craig, thank you again for all the work you've been helping me with Ecosphere Sciences. And Ken, as always, much appreciated for the hard work you put in behind the scenes and making this all happen. So thank you, guys.
Yeah, thank you.
You bet. And
Take
guys,
care,
don't forget that these guys also had some podcasts. So you want to head over. That's the Spotify one and Apple Podcasts as well. So they're on most of the podcast software.
Applied Mycology, if you just search it on any kind of aggregator, it should show up. Leif and I put that out in 2020. Was it 2020 or 2021? We worked on it at the end of 2020. In 2021, we released it. It's 10 episodes, and kind of instead of doing the unrelenting, endless march, we really found a number of people that we knew because it was focused for like one topic that are in the mycological community. We have a couple things recorded. We might release a second season, but kind of like, it's kind of like the kind of cult following, like get a follow up like maybe 5 years
maybe
later.
we can get you guys on here doing it, and we'll form the cult over here, and I'll do the back end. Anyway, guys, we're gonna end the show there. We'll have these guys back on definitely, Hopefully, you know, Leighton study group will keep going, and maybe they'll just come study over here for a while.
You never know.
You never know.
Appreciate